The last enzyme that's different in the gluconeogenesis pathway is interesting because it only is in the liver and the kidneys. This particular enzyme is glucose 6-phosphatase, and this is required to remove that phosphate group from glucose 6-phosphate to make glucose. In this reaction, that phosphate ester hydrolysis is going to be the driving force of the reaction. So again, creating that inorganic phosphate is going to be that driving force. This is going to involve a nucleophilic attack from the nitrogen of histidine, which will go ahead and form a phosphohistidine intermediate. So while there's no actual covalent bond between the histidine and the entire substrate, it is basically going to take away that phosphate group. As mentioned before, the enzyme is absent from muscle and brain, but it is, again, in the liver and the kidneys. So your muscle and brain, which are the ones that are going to need that glucose, can't actually get to that last step. So it's only the liver and the kidneys that can actually do this, which would allow glucose to leave the liver and the kidneys. This enzyme is associated with the endoplasmic reticulum. So whenever we're talking about where some of these are, so this enzyme is actually taking place in the endoplasmic reticulum. So in order to actually get this to work, it would be inside of the ER. So glucose 6-phosphate is going to enter via a particular transporter. So we talked before about how that phosphate group on glucose 6-phosphate is not going to allow it to cross a membrane by itself. But if there's a transporter, then it goes through just fine. It's going to use a particular transporter to then go from glucose 6-phosphate to glucose. And there are other glucose transporters to allow it to exit the ER lumen back into the cytosol, and then can also then exit the cell, so it can enter the bloodstream and get to where it needs to go. We've seen this slide before, so this is again just to remind you that we have glycolysis and gluconeogenesis, and they're essentially opposite cycles with particular steps in the pathway that are different and unique. The thing that when we started this we talked about was those thermodynamics, that you have to have something in glycolysis that's going to be that driving force, and the nice thing is that being a catabolic reaction, there's a lot of good exergonic reactions to drive glucose forward. And gluconeogenesis can't just be the reverse. So if it was just the reverse, you would have that positive 74 kilojoules per mole in gluconeogenesis. So we need something to help drive gluconeogenesis forward. And a lot of what that is there's additional molecules of ATP and GTP that are used in gluconeogenesis to be that driving force. So realize that there's 6 total nucleoside triphosphates, 4 ATP and 2 GTP, to get those 2 pyruvate molecules to go all the way to glucose. If we were just reversing glycolysis, we would only use 2 ATP molecules. So realize there's that net of 4 nucleoside triphosphates that are helping be that driving force of gluconeogenesis to get this to actually go all the way back to glucose. This is again just that reminder that there's 3 main reactions of glycolysis that are replacing gluconeogenesis. So remember that it's that weird thing that in gluconeogenesis that you actually have 4 enzymes, but it's replacing 3 main steps. Because at that very beginning, you need both that pyruvate carboxylase and phosphoenolpyruvate carboxykinase to take the place of that one glycolytic reaction of pyruvate kinase. I don't know if you've realized this, but your liver is awesome. That's basically what this is about. Is when your muscles go ahead and use up glucose in glycolysis, and if they're doing a whole lot of exercise and it even makes lactate, your liver will actually go ahead and reoxidize the lactate to pyruvate by liver lactate dehydrogenase, and then your liver can go ahead and continue in gluconeogenesis to go ahead and make glucose again for the muscles. So the muscles basically just get to use all the glucose that they want, and the liver kind of is that cleanup crew. Along with this is that your NADH can also be reoxidized during that reduction of pyruvate to lactate. So your liver basically helps kind of clean up after the muscles have used up all of the good glucose. This step, this notion that the muscles are going to use things and the livers are going to regenerate it for us is known as the Cori Cycle. So this is mostly here just to help with where everything is. So I'm going to have that problem with where my liver is and where my muscles are. I know where my muscles are. That one's easier because they're in lots of places. But what this does usually mean is that since you have a lot of glycolysis happening in your muscles, that means that you're going to have a fair amount of pyruvate going to lactate and producing that NAD plus. So then in the liver, you're going to have that opposite where you're trying to go from lactate back to pyruvate, back to glucose, and producing that NADH. So you also have the liver helping regenerate not only glucose, but also that NADH. For regulation, so this is just a summary of some of the regulation that we've talked about before. So whenever we talk about reciprocal control, that's basically saying that one cycle is inhibited while the other one is active. So whenever we say reciprocal control, we're basically getting double duty out of a particular molecule. So you can have one molecule that is simultaneously saying, not this one, yes to that one. Without that reciprocal control, you would have a futile cycle where basically things would just keep going around and around and around. So you have to have something to say, we don't need one of these, we only want the other one. So we want to make sure there's a tight control over which pathway is happening. That reciprocal regulation is going to depend heavily on the energy status of the cell. High energy is going to usually give us more towards gluconeogenesis. Low energy is going to give us more towards glycolysis. So again, energy status is low, we're going to degrade glucose to provide ATP. When energy status is high, we go ahead and make glucose and hopefully store that into glycogen. So when glycolysis is on, gluconeogenesis needs to be turned off. So this is just kind of try to look at everything all together about where things are. So we do have that glucose 6-phosphate will inhibit hexokinase. Realize that that is a substrate concentration and not necessarily an allosteric control. So that's why it says substrate level control on there, is because that's where that one happens. Looking closer at things like pyruvate kinase and that pyruvate carboxylase is that acetyl-CoA is going to inhibit glycolysis and stimulate gluconeogenesis. So this is going to be, again, one of those reciprocal controls. AMP, again, low energy status, so is going to stimulate phosphofructokinase. So if we don't have enough energy, we're going to try to produce ATP from glycolysis. If we have low energy, we don't want to be spending our energy making glucose. AMP is going to have that reciprocal control over phosphofructokinase and fructose 1,6-bisphosphatase. Fructose 2,6-bisphosphate is again going to have that reciprocal control. And you see that same reciprocal control with citrate. In this case, this is a TCA cycle intermediate. So essentially, when citrate levels are high, that means that your citric acid cycle has a lot of stuff in it. So it's basically kind of like a backup pipeline. Like, oh, we've already have enough of this, so we could just go back the way we came now. And we could go ahead and have gluconeogenesis happen because we have so many TCA cycle intermediates. Whereas if that citrate level is low, that means we've got a lack of TCA cycle intermediates, so we want glucose to keep being degraded so that we can go ahead and fill those TCA cycle intermediates. We're going to talk a lot more about that cycle after this next exam, and there's a lot of other pieces of that cycle that go off to make other things for the cell. So citrate is more of that signal that we need stuff that either comes out of glycolysis or we have enough of the stuff that comes out of glycolysis. That, my friends, is it. We made it through gluconeogenesis. Yay! I highly recommend you guys take notes on this and write things down. It's very helpful to put pen to paper and see if you guys can kind of recreate what's happening here because there's a lot of moving parts to all of this. Good luck!